Alright, hi guys. We are starting chapter 4 today. We will be working on section 1, which is cellular level of organization. A detailed study of the cell began in the 1830s. Scientists began working on a unifying concept in biology. It originated from the works of two biologists, Schleiden and Schwann, who started working in the 30s. They each came up with a portion of the cell theory working independently of each other. Schleiden was a botanist and he worked with plants, and Theodor Schwann was a zoologist who worked with animal cells, and they both arrived at the conclusion about the same time, independently of each other, that organisms were made of cells. Another part of the cell theory is that cells only come from pre-existing cells. Where that first cell came from, scientists aren't really sure yet. They're still working on it. And the last part of the cell theory, cells are the basic units of structure and function in organisms. So if you have a structure that is smaller than a single cell, it is not a living thing. Here we have some pictures of cells. We have a plant here. This looks like a lilac, and then a zoomed in view using a microscope. This is 80 times magnification. You can see the various types of cells there. On the right, we have a bunny rabbit and a zoomed in view of its skin cells. If I were to take a guess, this is at 59 times magnification. Here we have some organisms and non-living things here at the left side. Just comparing the size, we have atoms down here at less than a nanometer, all the way up here to a blue whale, which is somewhere between 10 meters and 100 meters. They get to be very, very long. We also see what type of instrument is necessary to see each of these objects. Down here from atom to human egg, you would need an electron microscope to get a clear view. You can also use a light microscope for chloroplast to frog egg. Okay, you get a good picture of what's going on there. And then whenever you get larger than a human egg, you can see that with your naked eye. Cell size. Cells range in size from 1 millimeter down to 1 micrometer in diameter. They're very, very, very small guys. Cells need a large surface area of plasma membrane to adequately exchange materials. They need a lot of space for things to get in and out. The surface area to volume ratio requires that cells be small. Large cells, surface area relative to volume decreases, which also decreases the efficiency of transporting materials in and out of the cell. So imagine Walmart, okay, very big corporation, very big building, lots of things going in and out. If they only had one register for you to purchase stuff, it would take forever for things to get in and out. If we, however, increase the number of cashiers, or number of registers, we increase the speed at which things can get in and out. So a large cell would be like one register open at Walmart. A small cell would be like having 50 registers open at a time. The larger surface area to volume ratio is advantageous for exchanging molecules, for things getting in and out. Here we have a graphic showing surface area to volume ratios. If we have one large cube, the total surface area is about 96 centimeters squared. The volume is about 64 cubic centimeters, and the surface area to volume ratio is 1 to 5. I'm sorry, 5 to 1. If we were to cut this one cube into 8 cubes, our total surface area increases, our volume remains the same, and our ratio is now 3 to 1. If we were to cut each of those cubes again into 64 cubes, our total surface area is increased greatly. Our total volume is the same, but our ratio is much larger. It is much more efficient for cells to be this size rather than this size. It's easier for things to get in and out. If there's something bad in this large cell right here, it's going to take a long time for the cell to get rid of it. But if it's down here in the smaller one, it's got a lot less area to travel to kick it out. All right, moving on to microscopes, compound light microscopes. Well, light is passed through the specimen. It is then focused by a series of glass lenses, and then it forms an image in the human eye. The maximum magnification is about 1,000 times. The compound light microscope resolves objects separated by 0.2 micrometers, which is about 500 times better than the human eye. And that's assuming that the resolving power of the human eye is 1. This is an image of a compound light microscope. We're going to be working with them tomorrow, so don't forget to wear your tennis shoes. Moving on to electron microscopes, there's a couple types that we're going to talk about. The transmission electronscope, abbreviated TEM. Electrons are passed through the specimen, and then they're focused by a set of magnetic lenses. From there, an image is formed on a fluorescent screen that looks a lot like a TV screen. From there, the image can be photographed. This provides greater magnification than the compound light microscope, and it resolves images 100,000 times better than the human eye. This is what a transmission electron microscope looks like. Here we have an image created using one of a euglena, which is a single-celled organism. Notice all of those structures inside. That's a lot of detail that we wouldn't have been able to get using just a plain light microscope. Another type of electron microscope is the scanning electron microscope, abbreviated SEM. Using an SEM, the specimen is sprayed with a thin coat of metal. Then an electron beam is scanned across the surface. The surface metal emits secondary electrons. Those electrons are then detected and focused by magnetic lenses, and an image is formed on a screen similar to that of a TV screen, and then it is photographed. So here is an image created by a scanning electron microscope. You can see the outside of structures using the scanning electron microscope, whereas you can see the insides using the transmission electron microscope. So the setup is very similar of the scanning electron microscope. So just take a look at that. I think that that's a really cool image myself, but then again, I'm weird. So moving on, magnification, resolution, and contrast. Magnification is the ratio between the size of an image and its actual size. An electron microscope magnifies objects hundreds of times more than a compound light microscope. The resolution is the minimum distance between two objects that allows them to be seen as two separate objects. A microscope with poor resolution would let a student see one cellular granule compared to two granules with greater resolution. Contrast is the difference in shading of an object compared to its background. Fluorescently tagged antibody molecules can help visualize subcellular components like specific proteins. Illumination, viewing, and recording. Light rays can be bent or refracted and focused as they pass through glass, but electrons cannot. Electrons have a charge and can be focused by electromagnetic lenses. The electrons leaving the specimen are then directed to a screen or photographic plate, which is sensitive to electrons, which can then be viewed by humans. Confocal microscopy, a major advance in illumination. A narrow laser beam is scanned across a transparent specimen. The beam is focused on one very thin plane in the cell. The person using the microscope can optically section a specimen by focusing up and down. Sections are made at different levels. This allows assembly of a three-dimensional electron image on a computer screen that can then be displayed and rotated on the screen. This can give scientists a better view, and they can manipulate the images much more easily using this technology. Video enhanced contrast. The microscope image can be recorded by a TV camera. The video enhanced contrast I have trouble with that word, guys, sorry. A TV camera converts the light image into an electronic image, which is then entered into a computer. The computer then edits it slightly, makes the darkest areas of the image darker and the lightest areas lighter. The result is a high contrast image, which can then be manipulated for greater contrast. So it's an easy way for scientists to manipulate the images to get a better look at what is under the microscope. Here is the graphics comparing a light microscope to an transmission electron scope and a scanning electron scope. So it gives you an idea of the different types of images created by each type of microscope. Here we have some photos of cheek cells using different mediums, different contrasts. I know in your notes it's black and white, so take a few minutes, study these. I think these are pretty awesome images. So just take a minute to look at those. And then we're done for the day, guys. If you have any questions, feel free to shoot me an email or ask me any questions tomorrow in class.